Dr. Dimitra Zotos is a Senior Research Officer at the Department of Immunology and Monash University. Thank you. And she's going to be our next presenter here. Dimi's had more than 15 years of research experience in immunology across many industries, academia, and both basic and translational research. We've worked with Dimi over many years with lots of research that she's done with our lung transplant service too. So without further ado, I'll get Dimi up here while I try and load Dimi's slides.
It's great to be here to talk about some of the research we've been doing. I'm excited to share this with you. My first slide is probably not needed in this audience. I'm sure many of you are aware as to why someone might need a lung transplant. It's more to point out some of the complications that occur after, which I'm sure some of you are familiar with as well. But the one I'm most interested in is chronic lung allograft dysfunction, or CLAD, or chronic rejection. This is the leading cause of complication and graft failure after the first year of a lung transplant. So the reason we're interested in this is because there's a real unmet need. We don't know which patients following their transplant are at risk of developing CLAD with any certainty, and there's currently no real therapies that can treat CLAD. So there's a need for us to find biomarkers to help in predicting who might develop CLAD, and also to potentially find ways to treat CLAD to increase the longevity of every graft that's out there. So the interest in my research is focusing on the immune system's involvement in this. So here in this slide, I'm just highlighting in the picture different parts of your immune system that are found throughout your body. The immune system is really important for recognising anything foreign and fighting infections. And unfortunately, in the transplantation setting, that is a foreign lung that comes into your body. And that's why you have to use immunosuppressive drugs to try and quieten down your immune system to prevent it seeing the graft that's coming in. But it's a fine balance. If you give too much of those drugs, you're at risk of more infections and other complications. So to date, the immune system has been significantly implicated in the development of CLAD. The degree of matching that you have between donor and recipients is important, and this, if you like, sets the sensitivity for if your immune system will see it or not. And Lucy's just spoken a little bit about that, and HLA. Antibody responses, if you have any pre-existing antibodies that might be targeted to your graft, or if you form any after your transplant, which Lucy was also talking about in her presentation. This means you're at a higher risk of developing CLAD, and how many infections you get after your transplant also influences your risk of developing CLAD. So the more infections you have, the higher the risk of developing CLAD over the term of the graft. The way we look at the immune system is by using blood. So all the cells of the immune system can be isolated from blood that we collect, and in the project work I'm showing you today, that's what we're focused on. So we use blood to read out on the immune system's activity and what might be going on to try and work out what's behind CLAD development and what we can do to try and intervene. So a vial of blood is taken from a patient, kindly donated to our studies, and we take it back up to the lab and we process it in a number of ways to isolate out the immune cells. So here in the tube, I'm depicting where we find all the immune cells, which are called peripheral blood mononuclear cells, or PBMCs. And in that collection of cells, there are two really important cell types, which are called lymphocytes, that are really important for fighting infection and keeping us healthy. They're B cells, which you briefly heard about from Lucy, which make antibodies. And there are T cells, which raise the alarm about foreign things that are going on and also help keep us healthy and fight infection. So together, these immune cells really keep us healthy. And in a transplantation setting, they could also be targeted towards the transplant itself and create some problems. The way we look at these in the lab is we run these blood immune cells through an instrument called a flow cytometry machine. You don't need to worry about the detail of what's shown on those plots or graphs there. It's more to illustrate the power of what we do. So using a flow cytometry machine, we can look at thousands of cells per second and read out different things about those immune cells. So every dot on those plots represents a single cell. So there's thousands of cells that we're looking at there, and we can read out different things about the B cells and the T cells that are present. So this is the approach I've used in our work, and for the project, our hypothesis or the idea behind what we were doing was that we proposed that the different lymphocyte populations that are present in the blood, B and T cells, looking at those and characterising those, we would be able to use that to predict who's at risk of developing CLAD. This is the concept behind our project. And before we could do the project, we had to have a biobank of samples to look at for the project. So this is what's depicted here. Between 2018 and 2020, patients were recruited into the study, and over a 3-year period they were monitored and clinical data collected. They were monitored for CLAD development, and in the first year we had blood samples being collected from pre-transplant and in a number of time points indicated there to 12 months after transplant. So from this patient biobank that we created, we selected out a group of patients to focus on. We found that we had 13 patients who had developed CLAD in the 3-year period, and we had a group of patients that hadn't developed CLAD. So by comparing these two groups of patients, we're hoping to reveal something about the immune system and a way to help further understand what's going on in CLAD. So as I said in the last slide, we take the blood, we process it to isolate out the immune cells, and through the flow cytometry machine, we look at the B and T cells and we get a whole lot of data that comes out the other end. This project was quite large. It ends up with us having over 400 data files to process and look at from the flow cytometry machine. And so we used computer algorithms and forms of artificial intelligence or AI to process and really analyse this data in a high throughput and a novel way for the field. So what did we find? I'll just orient you on my graph. So across the horizontal axis, I've got the times from transplant, with 0 being the prior to transplant time point, and vertically I'll be indicating the B cell counts that we saw. So that's the immune cell that's really important for making antibodies. And on the graph in the grey, you'll see those patients that didn't develop CLAD, and in orange you'll see the patients that did go on to develop CLAD by 3 years. So what we can see is that by the 2 stars indicated on the far right, that the 2 groups were different in their B cell counts overall, and specifically at the pre-transplant time point and then the 3 time points after that, there were less B cells present in the patients that went on to develop CLAD compared to those that didn't. So this finding was a surprising finding to us because B cells in transplantation in some ways are considered bad because they make antibodies and they can make antibodies to your graft. But in this case, we're seeing that if you don't have enough B cells going into your transplant, this can also present a problem that seems to lead to CLAD. So it was an interesting finding, and we had to step back and think about what is it about B cells and what could they be doing in transplantation that could be helpful, and how does this link to CLAD? So there were quite a few things that came to mind, and a lot of this is still ongoing research. This is kind of a new finding we've had. But the first thing we considered was what immunosuppressive drugs were the patients on, and was this the reason why we're seeing less B cells in some patients? And the short answer to that is no. There were no difference in the drug regimes that the patients were on that could explain our result. We considered whether B cells would be directly involved in developing CLAD, or if perhaps they were reduced for an unknown reason, but it was a good readout for predicting CLAD. And this is still part of ongoing work. It means the cells are just a bystander. We're still trying to get at that. We don't think that's the case at this point, but it's still under investigation. The other thing we considered was, are the B cells surviving okay in these patients? Is there some reason why the cells are not surviving? And so we looked at a key survival factor called BAFF in these patients, and we didn't see this to be the reason why there were less B cells in the patient that went to develop CLAD. B cells, their major role, as I've said, is to make antibodies, but there is out in the field proposed that there is a small population of what are called B regulatory cells present, and these cells are important for dampening down immune responses, and it's possible that this might be the population that's affected in our patient cohort, and this is still part of ongoing work to look at that. To date, the thing that we've been able to look at most closely is infection. So we considered that if someone doesn't have enough B cells, they can't make enough antibodies, and they can't fight off infection properly. They might be undergoing more infections because they don't have enough B cells, and maybe this is why our patients are more at risk of developing CLAD. And related to that is the type of antibody that can be produced. So we went to the clinical data and had a look at the infection rates that the patients experienced in the first year after their transplantation. And we saw that in the patients that went on to develop CLAD, these patients actually experienced more infections in the first 12 months compared to those that didn't go on to experience CLAD. So what does that mean, and what does that feed into our story so far? So I'll give you a little bit of a summary as to what we think is happening. When you go into your transplant, if you have too few B cells and you have your transplant, we think you're going to be more at risk of getting infections after. And if the more infections you have, this could be increasing your risk of CLAD. So this story is by no means finished and closed. It's part of ongoing work, but to date, this is the best clue as to what we have is going on, and there's probably more to the story. And this is the first time this result has been found in the field, and it does need to be looked at again in what we call an independent cohort. So we need to look at another group of patients and show that this is to be true. But what can we do with the data we have to date? We can use the data to what we call create a model to calculate the risk of someone having CLAD. So we can use the B cell count at the pre-transplant time point and feed that into mathematical modelling and create a formula for calculating the likelihood of someone developing CLAD. And so this is the formula here. There's no need to worry about the detail of what's shown there, just to say that it can be done. And what this would mean is if you had a B cell count, you can feed it into the equation and you get a probability or a likelihood that you may go on to develop CLAD within the first 3 years of your transplant. So here I've just shown roughly what that would look like. So patients with a B cell count in that red count area, so below about 80, have around about a 70% risk of developing CLAD within the first 3 years. And then if your B cell count is much higher, up towards 200, your risk is very low, but less than 1% using the data that we have to date. So as I said, we need to go on and validate these findings, and this is still very much ongoing work. And with that, I'd just like to acknowledge everyone that's contributed to this work, especially my PhD student Rahia, who's just submitted her thesis, and especially acknowledge the patients for donating their samples to this research, and Lungitude for supporting me over the last year, which has enabled me to do some of these experiments, attend a national meeting, and also prepare all this work for publication. So thank you very much.